Chapter 5, Antibody Structure and Function. So we'll go over what are antibodies, what are the general characteristics of them, what do they actually look like, the structure, all the different classes of antibodies, what actually is immunological memory, what are the specificity and diversity of each of the antibody classes, the genes that are actually involved in production of immunoglobulins, and what are monoclonal antibodies. So let's start off by what are antibodies? Antibodies are actually called immunoglobulins. When we did protein electrophoresis, which we'll be doing, and I'll show you here in a picture in a minute, there is an albumin, there's alpha globulins, beta globulins, and gamma globulins. And the gamma region is actually where the immunoglobulins reside. Just because it means immune proteins, basically is what that's saying. So that's why it's called antibodies, or immunoglobulins. So, immunoglobulins, antibodies, synonymous. So they're actually found in your serum. They're glycoproteins. As you see, a good chunk of them is actually a protein, a polypeptide, and a smaller percentage is actually a carbohydrate. So there's 5 major classes of antibodies that we have out there, and they all start with Ig, which stands for immunoglobulin. So it's IgG, IgM, IgA, IgD, IgE. Alright? So they are all part of what's called the humoral immune response. Humoral immune response refers to the antibody production. So we're talking about B cells is what we're dealing with here. Cell-mediated is more of a T cell kind of thing. So we're going to be focusing more on the B cell side. So, excuse me, as you can see from the electrophoresis here, and we'll be doing protein electrophoresis so you can see it. So here's the albumin, which, all of the blue stuff here is total protein. So what they do is we get like a gel, an agar, and we make a little slit in it, and we put the person's serum in it. So that comes from clotted blood, the liquid from serum. And then we apply an electrical current to it. So, and you can see here, this is the actual, what the gel would look like, and we stain it. So what this graphic representation is looking at the different colors that's on the electrophoresis, and you can see that the albumin is very dense. That's why it has a high peak. That's why you can see. So here's albumin, alpha 1, alpha 2, beta. There is a little bit of a beta there, and then the gamma here. The gamma here is where the immunoglobulins are, the gamma globulin portion. So this electrophoresis is actually done in an alkaline pH, and we'll be actually doing serum protein electrophoresis so you can actually see it. So the actual structure of an immunoglobulin is a 4-chain polypeptide, so that's called a tetrapeptide. So there's 2 large peptide chains, and you can see them in the dark blue here. The dark blue are the heavy chains. They have H on them because they're heavier in weight, basically. And then there's 2 lighter chains here, which you can see here on the side. They're actually labeled light chain. So they're lighter in color. So they're smaller polypeptide chains. Now, these are held together by non-covalent bonds and also by disulfide bridges, interchange bridges. And you can see the disulfide, the S is here, and the S is here, and here. And they call this the actual hinge pair region here. So the heavy chains, we actually use Greek letters for them, You can see like gamma, then we got alpha, mu, epsilon. So we go through that. The light chains are kappa and lambda. So they are the 2 light chains. So let's back up here for a second. So the light chains here are either kappa or lambda. And then the heavy chains, depending on which immunoglobulin class it is. If it's IgG, then they're gamma. Okay, so that's how that works. So basically the light chains are light in molecular weight, and here's the weight of them, but we're not really concerned about that, but just know that they're lighter than the heavier chains. And they're found in all the different classes, the 5 different classes we just talked about, the IgG, IgM, IgE, IgD, IgA. But only one type is present in a single molecule. Alright, so we have light chains, heavy chains. So light chains, light colors here, and then the heavy chains are very dark blue ones here. So there is a unique variable region that's on the what they call the amino-terminal end, which allows for particular bonding. That's this part right up here where the little asterisk is. So that's considered the amino-terminal end, the variable region, and that allows us to have specific binding to specific antigens. That's right. There is a different variable region for each antigen that's out there. It's hard to believe, but that's the way it works. Then in the carboxyl terminal end, which is more down in this area, is the actual responsible area for how the antibody works, this biological function. So there's 3 or 4 constant regions. They call that the carboxy terminal end. So the H chain between CH1, you see it labeled here, constant heavy 1 and constant heavy 2, you can see that there's actually a hinge there, and it's disulfide bonds that are there. That allows the little Y parts to sort of bend a little bit. So there are two, the very ends here can sort of like bend a little bit more so they can operate independently to bind to antigens, because this little top of the Y is what's going to attach to the antigen, to the foreign thing. Okay. It also assists in how complement will attach, in that this region down here. You can see complement and Fc receptor binding. And what that means is that when this top part of the Y attaches to an antigen, this is sort of like sticking out, so no complement can see this easier, and will allow complement to attach, and which eventually will kill the cell. And we got a chapter just on complements, don't worry about that yet. Also, it has Fc receptor binding. In other words, this little end part, if you remember in hematology we talked about mast cells, we talked about basophils and stuff like that. This little Fc region here, this is actually, especially of the antibody IgE, this is what the basophil will actually attach to in an allergy and allow the histamine to be released. So every part of the antibody has a very important part in what it does. Okay, and it's important for you to know all the different parts, because we'll be really going into those in a little bit more depth. So the carbohydrate portion of the immunoglobulins, that's found in all the classes, on all the different immunoglobulins. And that's usually located between the constant heavy 2 domains and the 2 heavy chains. So we're talking about in this particular area and here. Okay, and what that does with the carbohydrates is it allows it to move in the serum a little bit easier, so it increases the solubility of the immunoglobulin, so it can do stuff, so it gives us protection, so it doesn't degrade as easy. And then it allows for the Fc domains to be more functional, enhances them, allows them to do what they need to do. So what we do a lot of times when we're talking about antibodies is that we treat them with different enzymes. Papain's one of them, pepsin's the other one. So when you treat an when you treat an antibody with an enzyme, papain in this particular case, we actually will break it into 2 FAB fragments and 1 FC fragment. So we get that fragment of antigen binding and the fragment of constant. It's actually called the crystallized. That's where FC comes from. So you get 2 FAB fragments and 1 FC fragment. Now the reason this is important is when we start going into blood bank, we sort of have to understand what's going on with these antibodies. So that's why it's an important part right now. So, like we said, the FC fragment, very important part of what the antibody does. The FAB part is where we have the antigen binding. So pepsin, does it a little bit different. In other words, we only have 1 FAB portion here. So the antigen binding parts, the FAB parts, are held together still, but the FC portion is broken apart in non-functional pieces. So that's what goes on with the difference between papain and pepsin. Now also when we're talking about antibodies, we have what's called isotypes and allotypes and idiotypes. And that all refers to the amino acid sequence that's actually in the immunoglobulin that are part of the antigenic determinants, what we call how it responds to the foreign stuff, the epitopes, if you remember that from the previous chapter. So, and you can see with the different colors, they're sort of explaining it a little bit better here. Because remember, antibodies, that are produced by immunizations to, by a million different species produces antibodies. So we have to be able to have different types of antibodies for different species, for different antigens that we're dealing with. So it's amino acid sequence that will determine as to how we attach to this antigen, to the little FAB portions here at the top. And you can see each one's a little bit different. So the first one is the isotype. So look at the picture again. So here's the isotype here. Okay. So the isotype has unique amino acid sequences common to all the immunoglobulins of a given class. So in other words, all the IgG are all the same, all the IgM are all the same, so forth and so on. So they're identical in all individuals of a species, but they are different between species, like us and a dog. We still have IgG, but it's different amino acid sequence in them because, they react differently. Okay? And so all these amino acid sequences that give this IgG or IgM, so forth, are all located in the constant region of the heavy chains. So remember going back. Let's back up a little more here. Here's that constant region here, right through here. These two up here, see constant heavy 1, constant heavy 2, constant heavy 3, so on both sides. Okay. Whoop, went too far. So that's why, immunoglobulin G is a gamma, immunoglobulin M is mu, immunoglobulin A is alpha, then we got IgD, delta, IgE, epsilon. So it's all a bunch of Greek letters that are in there. So the 5 main different classes of antibodies. Now the allotypes means there's just a small variation in the amino acid sequence that can sort of present in individuals of the same species. In other words, my IgG might be just a little tiny bit different than your IgG. All right. So, and this is usually located just in the constant regions, especially of the IgG subclasses, which we'll talk about, and a certain part of the IgA, and also of the lambda, the light chain. So it's just minor variations. So isotype, allotype, and now we'll look at idiotype. So idiotype means there's variation in the variable region that give individual antibody molecules specificity. And that's usually located in the amino terminal regions of the heavy and light chains. So let's back up so you can see what we're talking about. So we're talking about right there. See where the different colors are? That's where we're talking about the different idiotypes. Oops. So, like I said, they're located in the amino terminal regions of the heavy and light chains. So that's in that FAB portion of the actual antibody itself. So, looking at an actual antibody, what are the different structures of antibodies? So you can see here, they're globulated, they're rolled upon itself, they're stabilized by a lot of disulfide bonds. They create more of a cylindrical structure called immunoglobulin. So it can fold. If you look at the front of your textbook, you can see an actual picture of an antibody there, that little orange or purple looking thing. That shows you what an immunoglobulin looks like. And because of this folding kind of stuff, that allows it to capture antigens easier. Because remember, the antigens are going to bind to that variable region, the FAB portion up there. That's what's going to happen. So it's going to actually attach to what they call CDR 1, 2, and 3. The CDRs up here. So IgG is the most predominant immunoglobulin in humans. You can see 3/4 of the immunoglobulins we have are IgG in nature. They are also the longest lasting of the immunoglobulins, 23 days. So it means in 23 days, half of them are gone. And then you got to go to, let's say half of 23, 16.5. Then it'll be a half again. So they last pretty long. And you can see they are a monomer. Here's the molecular weight and the sedimentation coefficient, which, we're really not looking at here. But it's not important for you to remember, which you're saying thank you. But there's 4 different subclasses, and you can see IgG 1, 2, 3, and 4. And of course, IgG 1 is the most common of all, followed by IgG 2. So these subclasses, you can see from the pictures, look a little bit different. So they have slight differences in their constant region, the amino acid sequence. They differ in numbers. Sometimes you can see the different pictures of it here. Position of where the disulfides are between the different lambda chains that are going on and the constant chains in here. So they sort of differ in function. So, but they still have in commonality all that stuff. Okay. So what are the major functions of IgG? Is it binds to complement. And you will find out, we talked a little bit about it in hematology. Complement is a series of proteins that can actually destroy cells. So one of the major functions of IgG is it actually binds to complement. Okay, so it leads to inflammation and destruction of foreign cells. IgG is the most efficient, followed by IgG1. So here's showing you IgG here. There's the antigen. Here's complement attaching. So it's allowing, eventually showing phagocytosis to happen also. But it's just showing you inflammation, destruction of whatever this foreign thing is trying to destroy. Other things that IgG is well known to do is it can neutralize toxins and viruses. I mean, that's real important right now. So here you can see antibodies here. Here's a natural killer cell because here's the antibody attached to this foreign thing because this is the target cell. It's antibody coded. That's that Y, that IgG on there. So what will happen is it's flagging to tell the natural killer cell to come and destroy it. So it can neutralize this virus. So it can neutralize toxins, So in other words, if you have, like your tetanus shot, it's actually antibodies against the toxin of tetanus. So that's how that works. Also, IgG is very important in that it crosses the placenta. It's the only one that's capable of crossing the placenta because it's the smallest of all of them. So that's actually what provides immunity to the newborn because that's all the immunity the baby has from the mother unless there's, breastfeeding involved. But that's a different antibody, but we'll get to that. But IgG is the only antibody that can cross the placenta. And that will be important when we start talking about blood banking there. Also, we use IgG in the laboratory to help us with laboratory experiments, testing, because it allows us to have precipitation and agglutination. And we'll get into that in another chapter. But there's different ways for us to test things. IgM is the biggest. As you can see, the molecular weight's pretty heavy. But it's only a small percentage. It's only 5 to 10% of all the immunoglobulins that we have out there. Its half-life is a lot shorter than IgG. It's only 6 days. So it can exist as a monomer on the surface of the B cells. Remember we were talking about that. We were talking about adaptive immunity. We had this antibody. It looked like just the Y of an IgG. But that's the monomer, and it's actually IgM because it's on the surface of a B cell at that time. But when it's in the bloodstream, it's actually a pentamer. So let's look at what that looks like. So here it is. It's like 5 of the Y's together. And it's actually held together by this little red thing. That's called the J chain. That actually forms a disulfide bond that links all the adjacent amino acids. So it looks like a little star. So it's got 10 antigen binding sites. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it's a pentamer held by a J chain, which has disulfide bonds that link all the adjacent monomers together. Okay? Now IgM is very efficient in bringing complement, in other words, called complement fixation. In other words, allowing complement to attach to the IgM so that it can eventually destroy the cell itself. It's also really big in agglutination, in other words, where we see molecules come together so we can visually clumping. It also can neutralize bacterial toxins and viruses, but it's also very big in the primary immune response. In other words, when you first get exposed to, we'll say, a virus, the first antibody that is produced is IgM. So once your body goes through antigenic stimulation, this is the first antibody that will be produced. It's also the first one in an infant that's actually produced. So IgM will be made as long as the antigen remains present, but it's usually, very quickly goes away. So it's important for us when we see IgM antibodies present in a person of a known, whatever it may be, then that tells us there's an acute infection. For instance, okay, Lyme's disease. Everybody knows Lyme's disease, all right? So we check for IgM antibodies, and we check for IgG antibodies. So if they have a lot of IgM antibodies present against Lyme's, then we know that they have an acute infection going on. If IgG is present, that usually indicates to us that it was a prior infection, because remember, IgG actually will come on the secondary immune response, which we'll talk about here in a little bit. So that's how, when they say do antibody titers, that's why they do that, so they can look and determine, is it an acute thing or is it a chronic thing? If the IgM's elevated, then it's definitely an acute response going on. If the IgG is elevated, then it's one that happened, in the past, and you've gotten re-exposed to it. So that's why they do titers, so we can see what the infectious status is of whatever's going on in the patient. Now, IgA is, you can see, only 10 to 15% of the immunoglobulins. Most of the serum IgA is a monomer with 3 constant regions, and you can see the molecular weight. There are 2 different subclasses of IgA, 1 and 2. So IgA1 is a very good anti-inflammatory agent. It actually can sort of slow down how IgG is initiating phagocytosis and chemotaxis. In other words, it sort of controls the inflammatory response, because we just said it's an anti-inflammatory agent. So it sort of keeps it in check, because you don't want stuff to go too overboard. IgA2 is actually found in secretions, and that's usually just on the mucosal surfaces. So like in your lungs, larynx, your genital area, intestinal tracts. In other words, any kind of port of entry that an antigen can get into, IgA is there. Because remember, IgM and IgG is located in the blood. So we want to try to be able to attack this foreign thing before it gets too deep into the body. So IgA is usually, because it's in the mucosal area, is called a secretory IgA. So it's usually synthesized by the plasma cells, which that's majority of all the antibodies are made. But these are usually the mucosal lymphoid tissue that's out here. So you can see here's a plasma cell here. So it actually releases the IgA, and it has that J chain that we just saw in the IgM. So the IgA and the secretory thing will end up being a dimer. In other words, 2 of them. That's what a dimer is. So it's 2 of them. So actually, as it goes, that J chain goes through and it gets ready to go to the epithelial cells out there, it will actually attach to this, what we call secretory component, that little green S-looking thing, before it goes into the epithelial cells. So you see it here. So the epithelial cells will sort of phagocytize, endocytosis. So it will stay in the epithelial cells, migrate its way through that mucosal cell, and then it will go out, be on the surface of the mucosal cells. So it will contain that secretory component that it got from the epithelial cells. So here's the dimer IgA with the secretory component and that little red J chain. So what are the functions of IgA? IgA is actually on patrol in all secretories. So it's the first line of defense. Remember, that's the portal of entry of a lot of different microorganisms. So we'll neutralize toxins, microorganisms. It prevents bacteria from adhering to mucosal surfaces, so we can try to knock it out first. I mean, when you get sick, don't you get like a stuffy nose or your intestinal tract gets, diarrhea, all that kind of stuff going on? That's what's there. It's trying to protect you. IgA is also located in the breast milk, so it will passively transfer stuff from the mother to the baby during breastfeeding. So it works extremely well there. IgA also acts as an opsonin. In other words, it attaches to this foreign thing, which then targets it so that the phagocytic cells can now destroy it. So an opsonin is just like telling something, this is what needs to be destroyed. So when you have aggregation of all these IgA immune complexes, that really triggers a different kind of pathway for complement. It's called the alternative pathway, because there's a classic and alternative, and there's actually a third one out there that we'll learn. But so it works in many ways. IgD is extremely scarce, and you can see a very tiny amount of the total immunoglobulins. It's a monomer. It's more susceptible to breakdown, has a very short life. Not too much is really known about it. I'm sure it'll play an important part eventually somewhere. So IgD, like we said, we don't know what it does. It doesn't really protect. It doesn't bind complement. It doesn't. And so what does it do? I mean, it's on the surface of the unstimulated, the naive B lymphocytes that are immunocompetent. So it appears after that monomer of IgM there. So they think it might play a big role in B cell activation right now. IgE, you can see, of the total serum, tiny amount. It's a monomer also, as a heavy chain, and you can see the Dalton weight. But the big important thing about it is that IgE binds to eosinophils, which then allows the enzymes into the eosinophil to release. So that will destroy, like, parasites, worms, that kind of stuff. Things that can't, are so big that the segs, the neutrophils, monocytes can't really break them, phagocytize them. So here's like another way for us to destroy foreign things. So IgE plays a huge part in allergic reactions. So you can actually see here's two adjacent IgE on a mast cell that is bound to a specific antigen, so that Fc receptor that we talked about, that the mast cell attaches. Because it attaches, it will degranulate and squirt out heparin and histamine, hence the red itchy area that we get. So in the primary versus secondary antibody response, you can see the difference between IgG and IgM, and the secondary antibody response, which is IgG and IgM also.